Hi, welcome everyone who's already there. Sorry that it took a few more minutes to start, but now we're ready. So welcome everyone. Hope you're having a great Tuesday, whether it's morning, afternoon, or evening for you. Today we have a talk by Margot Nelson from the Calvert Marine Museum in the US, and she'll be talking to us about the revision of the iconic Oligocene-Miocene dolphin Squalodon and the value of taxonomy in paleontology. So the format of today's seminar, if you're new here, we start with some welcome and announcements. That's what I'm doing right now. Then we have Margot's talk, will last around 30 minutes, followed by a moderated Q&A. If you have any questions, you can send them via chat to the questions at Paleoprix host, who today is Isaiah. You can already send them during the talk. And after that, Margot has agreed to stay for tea time, which is just an informal chat about work and her research after the talk itself. Then a little bit more housekeeping before we start. So Paleoprix values the participation of all folks interested in the paleosciences. And please remember to abide by our code of conduct during today's seminar. Also, please remember to mute yourself for the duration of the talk if you happen to find yourself unmuted, just out of respect for our speaker. And as I said, you can ask questions by chatting to the questions at Paleoprix host. Especially if you want to stay anonymous. If you don't want to stay anonymous, you can also drop them directly in the chat. Or if you want to ask a question yourself, you can also use the raise hand function. If you experience any technical issues during the talk, please also send them to the questions at Paleoprix host. Then we also have closed captions built into Zoom. You can either turn them on or off using the CC button. And finally, we are always looking for more speakers. Paleoprix only exists because we have so many cool early career speakers. So if you know anyone who could give a cool talk, or if you want to give a talk yourself, you're also very free to nominate yourself. Please go to the link that will be dropped in the chat shortly, where you can nominate someone. And then we'll also really appreciate it if you would fill out a weekly feedback form. This is anonymous, completely optional, and it's just for some demographic information for us to see if we can improve anything with Paleoprix. You can also find this in the chat window shortly. And with that, a bit more about our speaker, Margot Nelson. Margot did her PhD at George Mason University in the US, and she is currently doing a postdoc at the Calvert Marine Museum, also in the US. And with that, I would like to invite Margot to share her screen and start her presentation. I look forward to it. Margot, you're still unmuted.
Yes, okay. There we go. Yeah, thank you for the introduction, Nina. How do I collapse this? There we go. Cool. So thank you for attending my talk, either synchronously or asynchronously. This is largely based off of some of my doctoral dissertation research and some research I've been doing at the Calvert Marine Museum. And I'm a systematicist at heart, and taxonomy is really important to me. So I wanted to give a talk about my work and why we should still care about taxonomy in today's world. Will you not let me? Okay, there we go. So some brief stats about me as a person, as a researcher. I'm from Seattle, Washington originally, and completed my Bachelor of Science degree at University of Washington, which was really formative because there's a museum on campus. I didn't totally realize that paleontology was a career that actually existed outside of Jurassic Park. And going to a university that had a natural history museum on campus and being able to take courses and do undergraduate research really shaped my path. I completed my master's and my PhD at George Mason University under the guidance of Dr. Mark Ewan, who is a foremost expert in whale paleontology. So this was also like a very good place for me to be to develop my skill set. I have been conducting postdoctoral research at the Calvert Marine Museum, or the CMM, and my research focus is primarily on whales. I have an unhealthy obsession with whales. They're my passion, and my main research interests then are the evolution of marine mammals through a lens of anatomy and systematics. So little outline, I'll start with an introduction, which basically touches on why does taxonomy matter? Why does Squalodon matter? Methods, I will have a brief phylogenetic methods, but mainly talking about just the joy of collections research and the privilege that we have as paleontologists to go to some really cool collections. Some results, the taxonomy and phylogeny of this ancient whale Squalodon, discussion, and then what's up for me as a researcher next. So why is taxonomy important? It's basically the foundation of everything we do. We need to understand where something sits in terms of its phylogeny and its closest relationships. There's no paleoecology without knowing the species presence and their anatomy. You might know, okay, we have small dolphins, but that doesn't tell us a whole lot about the various ecologies unless we know the full diversity of all those small dolphins. We would have no diversity curves, right, without alpha taxonomy. And when we look at functional anatomy, we really need to understand where do different animals fit into their evolutionary context in order to see evolutionary trends. We can make some generalizations, but knowing precisely where these animals sit in is very important. So I am diehard on taxonomy. I'm very passionate about it. And the description of new species and the revision of older species is very important. Another reason why taxonomy is important is that it democratizes fossils. Without descriptions, you don't get quality photographs, you don't get detailed anatomical description. Sometimes people publish 3D models, and this all democratizes fossils because, like, you may never be able to go to certain museums, but through the publishing of specimens, you're able to see them nonetheless. So some terms for those who are maybe not as steeped in systematics as me, types are essential for communication. That's the other thing that makes taxonomy important is it allows us to communicate. A lot of advances, scientific advances, have just been the ability to communicate, like Linnaean taxonomy or the Rosetta Stone or Johnson's dictionary. We need to be able to communicate. So type species is the species upon which the genus name is based and will remain associated with that genus name during taxonomic revision. So the type species I like to describe as the thing upon which all other things are compared to. The holotype is that single specimen designated by the original describer of a species and available to researchers verifying the status of other specimens. So that holotype becomes that point of comparison to determine is something the species or not. In cladistics, that type species and the holotype becomes that that taxonomic unit that determines the contents of a monophyletic group. So having these sorts of things ironed out, really it's crucial for us to be able to communicate about diversity. So my research focused on Squalodon, which is a forgotten icon in whale paleontology. It famously to us whale paleontologists was the first illustration of a fossil cetacean was a specimen of Squalodon. The first named fossil-toothed whale was Squalodon. And Squalodon was initially thought to be a marine reptile until another paleontologist took a look at it and was like, no, this is a fossil whale. We have an idea of what Squalodon looks like. We have this platonic ideal, and Squalodon is a dolphin that's about medium size, and it has differentiated teeth along the tooth rows. So we have teeth that are split into something that resemble incisors, something that resemble premolars, and something that resembles molars. They also illustrate what we would call telescoping, which is the posterior migration of the nostrils. And their skulls are constructed very similarly to modern dolphins. They really occupy the space that's transitional in that they have this advanced degree of telescoping, but they have a very ancestral dentition. But based on the taxonomic practices of the 18th and 19th, the 1800s, it became a very messy group because people were just naming fragmentary fossils. So this is the holotype of a species of Squalodon called Squalodon vocantiorum. And it is not a good holotype. The only holotype worse than this one is a missing one. It's not diagnostic. And because it was such a dumpster fire, it was largely like forgotten in scientific research. It wasn't appreciated for what it could be. So when I started out, I was like, someone will surely revise Squalodon. And then by the time I was looking at doing a dissertation, no one had revised Squalodon. So it's just waiting there for me to reevaluate it. The implications of revising Squalodon have implications for the evolutionary trends, particularly in the evolution of teeth. So modern whales are homodont. They have a dentition that consists of basically conical teeth all along the tooth row. And I'm really interested in the progression of how this evolved. So the Squalodontidae have historically been a taxon that moves around in phylogenetic analysis. So one hypothesis puts it as part of the stem group of toothed whales, as opposed to the crown group, which is in this blue box. And so this would imply there's a single evolution of homodonty here at crown toothed whales. But I've highlighted this clade because there have also been hypotheses that the Squalodontidae belong in this blue box, along with some other heterodont toothed whales. And this would then imply that we have three origins of homodonty. So sperm whales here, they have their homodont. Then you would have, the ancestral condition here would be heterodont, and then another evolution of homodonty. And then you would have a third evolution of homodonty here. So parsimoniously, this raises some questions. Like, if this is the case, we want more evidence. And my hypothesis was that by revising the Squalodontidae, by including more of them in phylogenetic analysis, because this whole family in phylogenetic analysis has been historically represented by only one species, which is Squalodon calvertensis, was really hoping that we could solidify the position of this family within the phylogeny of Odontocetes, or toothed whales. So my research questions were largely what morphological features characterize Squalodon. So how can we distinguish Squalodon from all other toothed whales? Was Squalodon's distribution through time and space? So this was something that was somewhat up in the air. Because there were fragmentary fossils referred to Squalodon from all over the world, I needed to see if that was actually the case. What was Squalodon's real distribution? Likewise, there have been reports of Squalodon. There was one report of Squalodon from the Quaternary, but that was reworked. So what's its actual distribution through time as well as through space? How many species are there, and what defines those species? So, right, this is a question of like speciation events, diversity. It species count because there were so many like garbage species was really inflated. So what is the actual diversity of these groups, or diversity of this family, and how can we actually define those different species? What are their relationships in phylogenetic analysis? So by answering those basic research questions and getting those sorts of details ironed out, we can then start asking questions about what we can learn about dental evolution in toothed whales. Like, is there only one evolution of homodonty in toothed whales? Are there patterns in biogeographic dispersal within the Squalodontidae and beyond? is there niche partitioning? So we find multiple species of Squalodon in the same formation. Like, is there some sort of time element here? Is there division of niches? And from there, we can go down and down the rabbit hole of like functional morphology and stuff like that. So briefly, my phylogenetic methods. My phylogeny was based off of another person's work that I modified by looking at a lot of toothed whale teeth and coming up with characters that would potentially differentiate toothed whales based on their teeth. My matrix ended up having 104 taxa and 295 characters, which I then ran in TNT using like traditional parsimony with 1,000 replicates. I used a backbone constraint to force the relationships between modern taxa, since that is fairly stable for toothed whales. I ran it under both equal and implied weights. So these are weighting schemes that either treat all characters equally in terms of weight, and implied weights down weighs characters that have a high level of homoplasy. And if you have questions about phylogenetic methods and such, please ask me. I love actually talking about phylogenetic methods. And so I ran then analysis of support, namely jackknife and Bremer support. So a little bit of a travel log. I've been told my travel logs when talking about my research are fairly popular and enjoyable. And I got to do some really interesting traveling for this project. So I just wanted to highlight that. So I got to go to Brussels. This was to look at the holotype cast of the type species Squalodon great loopii. And one of the really special things about this museum was that they have this collection of iguanodons that are like beautifully lit up. They also have throughout their dinosaur exhibit taxidermy of birds, which was really delightful to have like this gigantic triceratops mount and then like these ducks next to it to, you know, tell us that, yeah, dinosaurs are still around. I got to go to this tiny little town in northwestern Germany called Brunda. I went there to study this beautiful specimen, Biosqualodon langevicii. It is a very, very small provincial town, and an American coming to this town was notable enough that I got to meet their mayor and I made their news. It was a really special experience. I went to Lyon, France. I went there to study this specimen, which was the holotype of Squalodon berriensis. This specimen's fantastically preserved. I actually got to see the middle ear bones. So all three middle ear bones are preserved on both sides, which was super cool. The museum looks like the Millennium Falcon. It was a really neat museum. And the other thing about collections research is you get to eat a lot of weird foods. One of the more baffling things I ate was called a French tacos. Tacos is singular. And it's halal meat and French fries and different sauces. I questioned my like choices, but I found out that that's a very like common Lyon street food. So it was actually like partaking in the culture. The last place I've been to for this research was Padua, Italy. This museum had just been renovated. They have one of the best collections of toothed whales from the Proto-Mediterranean. And this was a fantastic city to go to since it's renowned for its frescoes. So you would just go out to lunch and, you know, the town hall would just have frescoes in it. It was wonderful. Right. So now that I've gushed a little bit about how fantastic collections research is, a little bit about the results. So my first step was to take out trash. Right, the Squalodon today was this wastebasket taxon that had a lot of non-diagnostic fossils. So examples here of the various non-diagnostic fossils for which there were names. So for example, Squalodon hypsispondylus was named off of this single caudal vertebra. I've already shown you Squalodon boncontiorum, which was based on a single tooth. we have this notable example, which is a single tooth, likely of an archaeocete that's been reworked and put together. This was a holotype specimen. So this dramatically reduced the diversity of the family, the supposed diversity of the family, which I will show in the next slide. So here is the revised species count. So we can see, especially in the Burdigalian, which is the early Miocene, the species count was wildly inflated. It's much closer to 7, which is still a fairly large number of species for an Odontocete family, largely with a toothed whale genus. We only have one species. They're usually monotypic. And we only have one specimen. So this was step 1. The next issue that I dealt with was that the taxonomy of Squalodon posed a lot of issues. So there were 3 major problems. The first problem was, what even is the type species of Squalodon? So this was something that had been argued about in the literature for a very long time. And finally, another author went through the literature, like untangled all of these threads, and discovered that the true type species of Squalodon is Squalodon greatlupii, which has a holotype from France. It's pictured here in this illustration. The second problem, though, was that no diagnostic regions are preserved on this fossil. So for toothed whales, we largely care about the area surrounding the nostrils. We really care about the ventral surface of the skull, and we really care about the hearing complex, the ear bones. Those are the most diagnostic regions for toothed whales. So we don't have any of those regions preserved. So is there anything about this one specimen that could be really distinctive? And my findings were like, it's definitely Squalodon, but there's nothing really distinctive about it. And the third problem was that the original holotype didn't even exist. Another author stated that it was missing when he looked for it in 1923. And the International Code of Zoological Nomenclature, the ICZN, makes it very clear that a man-made cast can't serve as a holotype. So this was a major problem that I needed to solve. So my best solution was to find a appropriate junior synonym and to designate a specimen of that junior synonym as a holotype. So a junior synonym is a name that belongs to an already established species, or sorry, the specimen belongs to an already established species, but was given a new name. So I identified this specimen here, which was formerly Squalodon berriensis, went through the various species of Squalodon to try to illuminate, and came upon this as my best option since they were approximately contemporaneous. They had a reasonable geographic closeness, and morphologically they're nearly identical. So this solved that taxonomic problem, and now there's actually a really nice specimen upon which we can start studying Squalodon more closely. Other work I've done, right? So I looked at these species of Squalodon from the Proto-Mediterranean. Squalodon bellunensis had not been described since a monograph in 1916. So we redescribed that species, which has multiple specimens referred to it. So we were able to identify examples of intraspecific variation. We were also able to identify patterns of tooth wear that were unique to this taxon and not seen in other species of Squalodon. One specimen even had a deviated septum. You can see that. Let me see if my mouse can work. Yes, you can see that right here, that the bone that separates the two nostrils is wavy. So we're currently working on a monograph for that species. Squalodon peregrinus is another species that was actually never formally described. It was pictured in a few places, but it was given a name in a museum guide and has basically not been touched since. So again, we formally described this specimen. We made 3D models of it. We captured images of it. And this now makes that specimen a lot more accessible. We can start doing some more interesting research. So we were able to reconstruct the rostrum of this animal and make some paleoecological inferences about it now that we have better data for it. Another species that we redescribed was Squalodon languagii. This is the sister genus to Squalodon. And so for starters, the species was described in 1968. Very, very short description, was only photographed in lateral view, so we couldn't even get a full picture of the skull to study it. And also, parts of the specimen had actually been separated from the fossil before World War II and had been sitting in East Germany for decades, and were finally repatriated to the fossil after this original description. So we actually had a wealth of data that we could now access because the specimen was being revisited. We also found some really interesting paleobiological features, such as a really developed coronoid process. Here, this is for jaw musculature. It also has a really large temporal fossa, also for jaw musculature. And these features are not seen on any other type of toothed whale. Likewise, we can see a lot of attachment for neck musculature, and we even have a really well-preserved axis and atlas, so we can start to understand range of motion and bite strength and stuff like that for this species. Phylogenetically, this is the equal weights consensus of my trees, and we were really delighted to find that Squalodon today formed a monophyletic group. We were a little more disheartened to see that it didn't quite. So one specimen or one species of Squalodon did not group with the rest of Squalodon, despite having morphological traits that very clearly align it with Squalodon. And this becomes an issue of how much of your phylogenetic analysis do you want to influence by what you observe. So if you include characters to force relationships, like it becomes an issue of circular reasoning. But without adding any characters that we saw like as uniting the Squalodon today, we still got a monophyletic group, which is a really good sign. There are some interesting paleobiological implications in that our Mediterranean fossils formed their own small group. So maybe we are seeing individual speciation events, but we're not really seeing that in the rest of the tree. So this is an area of further research. So a little bit of discussion how systematics leads to advances. So firstly, I was able to revise the biogeography of this group. So the Squalodon today is restricted to the North Atlantic. This is an extremely restricted distribution compared to other toothed whales, especially modern toothed whales. Modern toothed whales oftentimes have like cosmopolitan worldwide distributions. Like if we think about the orca is found just about everywhere. Meanwhile, this one genus is constrained. And so this, for one, has questions of like, why do we not see crossover between Europe and North America? Why do we have these unique faunas for Squalodon on either side of the Atlantic? There's no biogeographic crossover. I'm also interested in whether or not a more endemic distribution is the ancestral state for toothed whales, and being cosmopolitan is something that they evolved later and is part of why some clades see huge success in the modern. In terms of phylogeny and evolutionary traits, evolutionary trends, my phylogenetic analysis revealed something really interesting. So we'll note that the Squalodon today and all other heterodont toothed whales fall outside of the crown group. So the crown group is all homodont, all with these conical teeth. They all look the same. And not only is that like just an observation that we get, it's actually we have dental characters that support the clade of crown toothed whales, namely the fact that they are all homodont. Um, so this was a really interesting result and says something about, potentially says something about the origin of homodonty that evolved once, which is a really interesting thing. The other thing that we discovered were patterns in dental conservation and how tooth patterning might happen in toothed whales. So something that we observed when studying the Squalodon today is that they have a very conserved tooth count. So the tooth count does not really vary at all. And this is unusual because modern toothed whales that are homodont can have varied tooth counts even between like individuals in a species. So one bottlenose dolphin might have more teeth than its sibling bottlenose dolphin. They really don't have any constraints in the number of teeth they can form. But it's clear that while they're still heterodont, they do have constraints. And we can also see that the teeth, right, we're not adding more incisors, we're actually adding more molars. And that says something about dental patterning as well, that we wouldn't be able to say without thoroughly studying this group. We can start talking about niche partitioning. So for Squalodon peregrinus, the oddball that I showed you earlier, we can see that it occupies a very different ecological niche based on its morphometrics. And we only really would be able to realize that if we studied it. And likewise, our descriptions of the squalodontids from the Calvert cliffs, the North American ones, we see niche partitioning by body size. So we have a more gracile, smaller Squalodon calvertensis and a very robust, large species called Squalodon whitmorei. And it's only now that we have a full idea of what species are present and what their traits are, that we can study these features. So some conclusions. Taxonomy is the most fundamental part of our science. It is needed for our communication, it's needed for understanding diversity and disparity, and it's needed so we can put each taxon in an evolutionary context. And revising Squalodon has had knock-on effects for larger themes of my research. So as I stated, right, I'm interested in whether toothed whales are ancestral endemic or cosmopolitan. And I'm interested in how homodonty arose in toothed whales. And in revising Squalodon, this has advanced my research in those areas as well. Finding new species is really important, right? We want to add to the diversity of life. But when we revisit these understudied and historical species, we can also lead to breakthroughs and lead to these species being studied in a new light. I know that my research on Squalodon has been able to unlock research into like biomechanical analysis now that we know where they fit in terms of evolution, and now that we even have like 3D scans of these specimens, which I was able to create. So I know that especially in the US, especially today, taxonomy and systematics are not seen as cool or hip or important. But keep your head held high. Like, it is actually a really important part of our science. So what's next for me? I am slightly moving on from Squalodon, even though Squalodon like forms 90% of my thoughts. So I'm also just really interested in looking at the evolution of skull shapes and trying to understand skull development. So I have a project going on where I'm looking at skull development in ancient dolphins and seeing if they have if ancient and modern dolphins share underlying patterns. So that's a new area of research for me. I'm also really interested in the evolution of skull shape as it attaches to climate. So in toothed whales, there is a like faunal turnover between the mid-Miocene climatic optimum and the cooling period after. So I'm really interested in quantifying that faunal turnover in terms of morphological diversity. So those are areas I'm exploring next. I thank people for their time and for watching my talk. I have a lot of acknowledgments. Thank you to co-authors, committee members, and like all the museum staff that helped me with these specimens. Um, and yeah, I'll take any questions you might have. Thank you.
Thank you very much for your talk, Margot. That was fascinating. I know from a lot of colleagues that are taxonomists, they share similar struggles. But me as a stratigrapher, I'm very happy to see that people are still doing taxonomy. And also really cool that you made it to the news in the little town of Bunde. That's really funny.
Yeah, it was surreal. Like
And that you
I
got to meet
remember
the
my like
mayor.
camp there at the museum. Yeah, being like the mayor's coming to visit. And I'm like, oh my God, what? Like I'm just a little PhD student. I'm know nothing.
Yeah, that's very cool. Yeah, a little reminder to our audience that if you have any questions, you can drop them in the chat or send them to our questions host if you want to ask them anonymously. We already have a few questions, so I will just start with those. The first one is, how about specimens in collections that might be currently mischaracterized as something else that might actually also be Squalodon? You think this is something that needs to be looked into, or has it already been looked into?
There are some places where there are collections that supposedly have Squalodon in them, that I'm really desperate to get to. But now that my research is out there, it is able to, and other researchers at these institutions are better able to identify whether the specimens they have are Squalodon. Um, like I've had a few leads from people in Australia, New Zealand, and in California, yet to fully study and determine if they are Squalodon. But yeah, like going through museum drawers is really important because you never know what's been labeled incorrectly. I'm currently involved in a project where I was brought on because they thought they had a bunch of Squalodon material, and then I looked through all the material and was like, this is not Squalodon, this is something completely different. Um, and so there's also the case of, yeah, like we can go through drawers and discover stuff that is Squalodon. Um, yeah, poking through museum drawers is super important. It's a super useful activity to make new discoveries.
Yeah, still a lot of work to do in museums, I suppose. I think also because there's no database, I guess. Another
Yeah, we have
question.
databases.
Oh,
Oh,
no,
yeah,
you can
we have
continue.
we have databases of like, so for example, the paleobiology database, that is based off of things that are only in the scientific literature. So museums collections really are like dark data. And so reviews of what's in museum collections, you know, and that get published, those can make it onto the paleobiology database. So even just like faunal analysis work where you're able to determine like the number of specimens and where they came from, even if they're not necessarily like a new species, like that's still really valuable.
Oh, that's good to know. So we have two questions now in the chat. I'll first read the first one that I asked, and then I'll move on. So the first one is, how do you deal with potential ontogenetic differences in the specimens?
Yeah, so that's a really good question. For whale paleontologists, there's a fairly good, we understand the sequence of suture closure in odontocetes. And odontocetes will actually like fuse most of their sutures in their skulls as they grow older. So that is one way that we're able to determine adulthood versus juvenile. And most of the specimens I did look at were all adults, based on like their patterns of suture closure. But that's one way that we would look for like ontogeny and be able to assess ontogenetic differences is if we saw an animal that had much more open sutures we'd be able to determine that they were younger. So that's part of what my research now I want to my sorry, can I not go back? Ooh. Um, so my research here, right, is to actually quantify how those sutures develop and hopefully give us a framework for understanding ontogenetic sequences in fossil dolphins as well.
Great, thank you
But
for
yeah,
your
we use it.
answer. We then have a question by Morgan Purefoy. I hope I pronounced it correctly. They ask, hello, thank you so much for your presentation today. What was the most fascinating incongruency or outlier you noticed between older specimens or phylogenies and the tree that you constructed yourself?
Let's see, let me go back. Um, yeah, that's a good question. Probably the thing that intrigued me the most is so when Squalodon was first described in the 1800s, it was really seen as like this like missing link from the ancestral whales like Basilosaurus to modern dolphins. And then starting in like the 1990s, there was like this huge blossoming of research that showed that there's actually many different types of kind of toothed whales that have an advanced degree of telescoping but are still heterodont. So all of these guys all kind of fit into that group. And one of my biggest surprises was that actually they created their own clade with supporting like with synapomorphies that supported them as their own unique clade. So to me, this was a really interesting result. It might suggest that there is like this diversification event from a common ancestor of derived heterodontocetes that appear from the late Oligocene into the middle Miocene.
Very interesting. Let's see, we have another question by Joseph Sullivan, who asks, great talk. Did you ever recognize a hybrid like a Noaluga?
So hybridization is like we know that whales can hybridize. It's very hard to know if that hybridization capability existed in the fossil record. Um, I didn't really see anything that was like a chimera of different traits. Each species was pretty easy to define as a unique species. So I don't think I saw any examples of sorry, I completely blanked on the word of hybridizing. There we go. But we know that is possible in odontocetes. Would love to know how far back it goes, if that's something that they can just do ancestrally, or if it's something unique to like these really rapid radiations of modern dolphin tooth whales.
Interesting, thank you. And Margot also, oh no, Morgan also says, thank you for your answer. And Joseph also says, thank you. So I think that's all clear.
Okay.
Morgan has another question asking whether any morphological characteristic noted that hinted at distinct behavioral traits in Squalodon.
Yeah, so I don't have a really great picture of it here, but in Squalodon peregrinus, right, we were able to do some morphometrics. It has a very short snout and very wide compared to other members of Squalodon. So most members of Squalodon look like this, where they've got a really elongate, narrow snout. And this animal has a much shorter snout, a wider skull. So we're pretty certain that this is occupying a different niche. Exactly what that is, we have yet to explore. But when we put it in what's the word? Like morphometrics, we saw that it didn't. The only places where it really clustered with Squalodon was in terms of like the tooth pattern, because it does maintain that same tooth pattern that all members of Squalodon have. But otherwise, is clearly doing something very different. The orbit is very different in this animal. The snout's very different in this animal. For Squalodon bellunensis, something that we discovered when we were looking at these specimens is that all known specimens of Squalodon bellunensis exhibit really heavy tooth wear that we don't see in other species of Squalodon where we have the teeth really well preserved. And because it's happening in like every specimen, we're able to say this is something that is, a behavior of not this single individual, but of this species. So we're currently trying to figure out like how to test these differences, but clearly it's doing something different from other members of Squalodon. So that's an area of further research and something that we were like able to discover just by reviewing these specimens, because it wasn't something that was really clear just based off of like the historical publications.
That's very cool. Then we have another question by Joseph, who asks, do you know of a book that you can recommend that deals with the evolution of whales?
Yes, so there are two really good books I can think of. One is by Annalisa Berta, who is like a famous marine mammal paleontologist. Let me quickly Google the name because I don't actually have it in front of me. It is The Rise of Marine Mammals, 50 million years of evolution. And so that goes over the evolution of all marine mammals. It's a very approachable book. It's not super long. And it covers the main details. A more in-depth book that's about cetaceans in general would be Cetacean Paleobiology. And that is like basically a textbook written about the evolution of whales. And that is probably those two are probably the best books I can recommend.
Thank you so much for the recommendations. That's very helpful. We have one more anonymous question. And they ask, do you know whether Squalodon originated closer to North America or Europe? Which of these populations might be more ancestral?
Um, so that's a really good question. And I think it comes down to um, understanding the distribution of Eosqualodon a little bit more clearly. So right now, the only specimen of Eosqualodon that we know of is in Germany. There's another species, Eosqualodon laterastris, but I'm more convinced that that belongs in Squalodon. That's a whole nother area of research that I didn't really have time to discuss. But there have been kind of Twitter accounts, which are of course the best scientific resource, of Eosqualodon in North America. So I think that would be an area of exploration for me next, to understand, really using the stratigraphy to try to figure out which Eosqualodon is older. That might give us an idea of which population is ancestral. But of course, you can deal with like maybe the North American Eosqualodon is more ancestral, and then you have Eosqualodon arriving in Europe, and then they originate in Europe and then go back to North America. Yeah, I'm not entirely sure where that would go, but that would be my next area, is to understand like the stratigraphy of these groups a little bit better.
That's very cool. Just a follow-up question for me. How good is the stratigraphic constraint usually for these, especially these like specimens that were collected in the 1800 somethings?
Yes, not great. For things on the North American coast, it's fairly well constrained. Um, we are getting we're getting better data for Italy. For the Italian fossils, are finally able to settle on an age of early Burdigalian. It used to be unclear whether it was Burdigalian or Aquitanian. But yeah, temporally, like the European ones are not super well constrained, and a lot of these specimens were found in quarries that are now closed, so it's not easy to go back and do more work. Like the quarry that Squalodon bellunensis is from is just like completely infilled now, so can't go back there.
That's unfortunate, but you just have to deal with the data that you
Yeah, and
have in
even
that
like
case.
yeah, but what was interesting, and I could have touched more on this in my talk, is that just by like redescribing these fossils, I was able to constrain the stratigraphy pretty significantly, because I was actually able to pinpoint where these fossils were really from, just by doing a lot of sleuthing. Like this was a project I think would only be doable now with Google, because it just required a lot of like Googling weird things.
Such as, if I may ask.
Um, so yeah, so for Squalodon great loopii, the neotype, there it is. There's my beautiful. Um, so the original publication is from 1861 and says it comes from a quarry owned by the Comte de Bourg. Um, and so I was like, okay, well that's very vague. Could be anywhere. They specified that it was close to a town called St. Paul Troy Chateau. Um, and so I basically was like, quarries owned by Comte de Bourg, and found where this thing came from. But it was like, yeah, something that I would not have been able to do really without the power of Google.
Yeah, I understand. To search in that for books is it's not
Yeah.
possible, at least in a timely manner. Um,
Yeah, and lots of thanks go to the Biodiversity Heritage Library. That is a phenomenal resource for these like older papers. Um, a lot of material I was able to find through the Biodiversity Heritage Library.
that's very cool that these resources are available. Okay, I have two more questions, but they're kind of one question, and I think after that we will finish.
Okay.
They're both related to the time it takes to analyze a specimen. So I've combined them here together. So the first one was, how long does measuring or analyzing a single specimen take? And then the second one is, thank you for the talk. Can you talk a bit more about the 3D scanning the specimens and making them available to others? It seems like an ideal
Yes.
way to digitize collections, but how realistic is it to do this for large amounts of specimens?
It really, so measuring and such, so it really depends on how much time you have and how many specimens you have. So like when I was looking at like the neotype of Squalodon great loopii, I really like spent, I think measuring and such was like maybe like it did not take me very long, maybe like half a day, but I was able to like, actually just sit there with the specimen, more carefully measure it out. Um, spend a lot of time with coding for phylogenetic analysis. So that is one thing that you do prioritize with specimens. Um, with things like ImageJ, like that can help measuring specimens a lot, especially these big tooth whale specimens. Like the skull of Squalodon languagii is like almost a meter long. So that wasn't something I would be able to measure with calipers. Um, but coding for phylogenetic analysis, that's something you really is best to do with a specimen right in front of you. So that takes up the majority of my time when I'm with a specimen. I spend, that can take up to a day. I also will spend a day just looking at the specimen and detailing its morphology. Like, so when we do anatomical publications, we like describe like the way I like to say it is that like a space alien should be able to reconstruct the skull based off of the photographs and your description of the anatomy. So I make a lot of notes about what I see, so that forms like the overall description. Um, when you're working with a lot more specimens, so like when I was in Padua, right, I had multiple specimens of Squalodon bellunensis, I had Squalodon peregrinus, and then I had Eosqualodon laterostris. I had to look at all of those within a week. You do have to speed up your processing of those specimens. For 3D scanning, there's a few different methods. So for Squalodon peregrinus and Squalodon bellunensis, my co-author was actually able to use like I think his iPhone and like photogrammetry to create 3D specimens. I've used a light scanner to create 3D models, and the speed at which it takes really depends on how much you practice. But creating a quality 3D model took me about half a day, just getting the scans. Unfortunately, so speaking of things that democratize fossils, so sadly my 3D scans for this specimen were actually the laptop that they were on was stolen. So I felt really bad telling the museum that I would have these 3D scans, then being like, someone broke into our $11 computer. But yeah, like I think that going forward, that should be our standard, is that you offer some sort of 3D model with described specimens. It isn't that hard to do with today's technology. Like I said, my co-author made very good scans of these guys off of his iPhone.
Thank you. I think that would be very cool to have in the future, 3D models. And everyone can contribute a bit to digitizing collections.
Yeah.
Yeah, with that, I would like to finish the Q&A. Thank you again a lot, Margot, for your time and your answers. Thank you also to the audience for your questions. Before we go into tea time, I will quickly share some details of next week's speaker. So thanks again, everyone, for joining us. Again, we would really appreciate it if you fill out the weekly feedback form in the chat so we can learn a bit more about our audience. And then next week, I hope you all join us again on December the 2nd, same time, on a talk by Laura LaRocca. She will be talking about glaciology. She's from the Arizona State University in the US. So to those of you that don't stay for tea time, I hope to see you then. And for those that do want to stay for tea time, so tea time is just an opportunity to have like a very informal chat with a speaker if you want to ask maybe some more detailed questions about her work, or maybe some things related to how you get a postdoc in a museum or something like this. So yeah, please stick around for a few minutes if that's something that interests you. Before we do that, however, I want to give our speaker a break. She's been talking for almost an hour, so we'll have a 2-minute break where you can maybe drink some water, turn off your camera, and after that, we'll see you for tea time, and also we'll stop the recording then. So those of you that stay for tea time, see you in 2 minutes. Those of you that are leaving, I hope to see you next week.